Hi, good morning, everyone. All of you are welcome. We are starting now the third virtual sections of the Xylella fastidiosa workshop from diagnosis to management. This workshop was organized in partnership between Exalc, Escola Superior de Agricultura Luiz de Queiroz, from University of São Paulo, and Citrus Research Center from Agronomic Institute, and it is supported by the CITAD, XFAS, Rede Ibero-Americana para a Vigilância de Xylella fastidiosa, and also by Department of Entomology and Acarology from University of São Paulo, and graduate program in entomology from University of São Paulo. The workshop is designed for researchers, professionals, growers, and students. And the main objective are to provide an updated overview of the plant pathogenic bacterium, Xylella fastidiosa, its importance, classification, host plant interactions, transmissions, detections, and monitoring, as well the management and certification strategies. For more detailed information on the program, please check in the workshop website that was posted in the chat. In this website, you can also find the YouTube links to the recording and previous workshop sections. About these specific virtual sections, you're gonna have two speakers today, 4 minutes each talk. Just pay attention that the debates will be afterwards, so the questions, no question during and between the talks, only during the debate. However, the audience can post their questions in the chat during the talks and debate. Take note or go or do the questions in debate sections, or write this question in the chat. Remember, you are remembered about the list of the audience. Google form will be posted in the chat. Please fill it. In these sections, you will have the opportunity to learn about important topics related to disease and bacteria diagnosis and novelties on Xylella-related disease surveillance. Again, take note about the questions, and at the end of the two talks, you will have questions and answers sections. Finally, starting with this first talk today, it's a pleasure to introduce the colleague, Dr. Marie-Agnès, that will talk about disease diagnosis. Dr. Jacques is an engineer. It's an engineer in agronomy, and she did her master science and PhD in plant pathology, both in France. Currently, she is senior scientist in INRAE and director of research working at the Research Institute of Horticulture and Seeds in Hungary. She has a great experience of research on plant pathogenic bacterium, teaching and training activities. Mad, it's a great pleasure to have you in this workshop as a speaker. The floor is yours. You have 4 minutes, and good luck.
Thank you very much, Elvesio, and hello everyone. It's really a pleasure for me to present some of our work today. And first, I would like to thank Elvesio and his colleague for inviting me to present our work today in this very interesting workshop. So I hope everyone can see my PowerPoint presentation. So today, I will first summarize some characteristics of this bacterium that makes it a real threat for agriculture and landscape, and characteristics that have impacts for diagnosis. So you may all know that the fastidious nature of Xylella, which is a burden for detection and more generally for in vitro, translates in planta in a long asymptomatic development between the infection time and the occurrence of symptoms. So this has two consequences for a diagnosis. First, the visual examination of plants is not reliable to evaluate infection due to Xylella, as latent asymptomatic infections can last a very long period of time. And second, the isolation is not mandatory to declare infection, as it can be very difficult depending on the subspecies of Xylella and depending on the host plant. Also, this bacterium is limited to the xylem of the plant hosts, in which it forms biofilms. So biofilms are aggregates, and the bacterium, when it's aggregated in biofilm within the xylem vessels, it plugs the vessels. And in response to the infection, the plants overproduce defense compounds such as tyloses, also pectins, that altogether led to an embolism and results in symptoms that are very similar to those due to anhydric stress, which means that symptoms are not typical and hence really not reliable to declare infection. So this bacterium is naturally transmitted among host plants only by sap-sucking insect vectors. And a wide range of insects are potentially able to transmit it from these various families. And as a result of human activities, the bacterium is also efficiently transmitted by plants for planting. And this led to intercontinental dissemination of the bacterium. So regarding infection and diagnosis, it is important to note that the bacterium colonizes two distinct habitats that need to be sampled. So the xylem network of the plants and the foregut of the insect. The symptoms that are caused by this bacterium, once it has reached a quorum locally in the plant, are very diverse. And this is a pain for diagnosis. The symptoms depend on the combination of host plant and Xylella subspecies or strain. As the bacterium invades the xylem vessels, it blocks the transportation of mineral nutrient and water. And more generally, symptoms are very similar to those of anhydric stress. Plant can present leaf scorches, wilting of foliage, withering of branches, defoliation, chlorosis, stunting, dwarfism, but also symptoms as different as yellow spots or irregular lignification of bark, crown dieback, or tree decline. And in some cases, it can lead to the death of the plant. So here you have various illustrations of symptoms due to Xylella, and among the main diseases that are caused by Xylella. Here you have typical symptoms of the Pierce disease on grapevine. Here you have, but you are more familiar than me with the citrus variegated chlorosis. And here you have quite mild symptoms of olive quick decline syndrome in southern Italy. Moreover, the symptoms that are due to Xylella are potentially symptoms that are caused also by abiotic and biotic factors. So there is a risk of confusion. So visual examination of plants, it's absolutely not reliable for evaluating Xylella infection. The host range of this species of plant pathogenic bacterium is really large, as more than 660 plant species have been declared infected by this bacterium, and most of them are perennial plants. But what is very important to have in mind is that not all of them declare symptoms and declare a disease after infection. Indeed, it is mostly supposed that most of them remain asymptomatic. So disease is not caused in all hosts. And currently, there is no curative method in field. Thermotherapy can be used only in the case of nursery vitis plant material. And there is nearly no resistant genotypes. Only a few tolerant genotypes have been described in the case of grapevine and olive trees. This highlights the importance to detect as early as possible this infection due to Xylella in order to eradicate the infected plant, in order to limit the dispersal of the bacterium and the dispersal of the disease. So in diagnosis, we are very lucky to have access to a standard published by the European Plant Protection Organization, the EPO, which describes a diagnostic protocol for Xylella-caused disease. This standard is frequently updated and serves as a reference for national plant protection organization in Europe, but it is available to everyone. So the 5th version of this standard is under finalization and will soon be available. The first step in diagnosis, after a visual examination of the plant or plantlets or plant material, is the sampling. 2 EPO procedures are dedicated to Xylella and relies on international standards for phytosanitary measures that are published by the Plant Protection Convention, IPPC. And a survey card proposed by the European Food Safety Authority also provides key elements for a risk-based and statistically sound survey design. So these are very key references that everyone needs to have access to when it is a problem of sampling plants or, nurseries to detect Xylella. So to limit the risk of false negative results, various points need to be considered. First, at introduction points. The most relevant pathway for the introduction of Xylella fastidiosa in a yet area that is free of this pest is the importation of plants for planting and infectious insects that originates from area where the pest is present. Dormant material that is imported from countries where Xylella is known to occur may need to be sampled and tested for latent infections, or they may need to be held in contained condition for inspection during the following growing season. And here, what is very important to consider for a risk-based and statistically sound survey, it is the size of the inspection unit, the lot size, and the confidence level that is desired, and the level of infection that is expected. And you have to bear in mind that the plant material may be most of the time asymptomatic. So at production places, taking into account that the distribution of the infection is not uniform in a site, the number of individuals, so plants or trees, that need to be sampled should be determined based on expected confidence level regarding the presence of symptoms. Similarly, it has been very well illustrated in the literature that the bacterium has an aggregated distribution in plant, which means that several organs should be sampled for an individual depending on the size of the tree. It has been calculated, for example, by Italian colleagues that 4 to 10 subsamples should be taken in olive trees depending on the age of the plant. The concentration of the bacterium in the plant also depends upon environmental conditions. It depends also on the strains and the host species or the cultivar. So to maximize the likelihood of detection, sampling should be performed during the period of active growth of the plant and during the period of activity of insects. So in all cases, sample materials should be organs or tissue that contain xylem vessels, because the bacterium inhabits those xylem vessels, which means that young twigs, leaf petals, leaf midribs, but also blueberry stem or citrus fruit peduncles can be sampled. Potential vectors can be also sampled, but this is not my area of expertise, so I will not provide a lot of details on insect samplings and analysis. However, you should be aware that recent papers were published that indicate that sampling of insects could be very effective in Xylella-free area to early detect the presence of the bacterium. This is quite important. It's a paper that was published by the team of Stephen Cornell in Warwick University. So when a lot of plant material should be sampled, to reduce analytical cost, it could be very interesting to pool material. And pooling of plant material, so which result in composite samples, has been studied, and guidance was provided by Italian colleagues, and this guidance are now included in the EPO standard. Here, what needs to be considered is the number of subsample per individual, as the percentage of subsample testing positive can be as low as 33%, depending on the condition, so species, symptomatic or asymptomatic plant, and so on. Then the number of individuals that should be sampled, the type of tissue, which depends on the plant species, and the volume of extraction buffer for subsequent analysis. But in a recent paper, Italian colleagues also add, so it is in a paper that was published in 2021, these Italian colleagues add also the diagnostic sensitivity as a function of the detection assay that was used, and they compared the efficiency of Harper's qPCR test, real-time LAMP PCR, and ELISA. And you can see here that most of the time, these different diagnostic methods are very equivalent and very efficient, but for some species, so for example, Herbaceous plantlets, the efficiency, so the diagnostic sensitivity, can be quite limited for LAMP and ELISA, and this is also the case for almond plant species. So the standard provided by EPO provides these very easy to read flow diagrams that are dedicated for plant sample or vectors. And they are they are quite clear and easy to hands-on. I have just summarized in this following slides a few important points that we have to keep in mind. So first, what is in contrast to most plant pathogenic bacteria, there is no recommendation for isolation. And to consider that a sample is infected by Xylella, we have two different cases, being in a pest-free area or in an areas where the pest is known to be present or in buffer zone. So the number of tests that should be applied is different. One test is sufficient when we sample in an area known to be infected or in buffer zone. But in pest-free area, two different screening tests should be run, and these tests should be based on different biological principles. So they should target different parts of the genome for molecular tests. And in case of asymptomatic material, only molecular tests should be performed. Serological tests are not sensitive enough. So this standard provides a list of various serological tests, so to be used only in a pest, in an area where the pest is known to occur, and also a list of various molecular tests that are available to detect Xylella. There are conventional PCR tests, real-time PCR tests, isothermal amplification tests. I mentioned here the qPCR test of Harper and colleagues that was published 12 years ago, and this test is considered as the most sensitive test and most reliable test so far. It is widely used throughout the world, and especially in countries where the infection by Xylella has not yet been detected. Regarding the identification of the subspecies, there are various conventional PCR tests or real-time PCR identification tests, but most persons rely on the sequencing of 2 to 7 housekeeping genes in a frame of a scheme that has been described a very long time ago by American colleagues, and it is very helpful and very portable. So now I will present 4 studies we have led or we were associated to in my team that aimed at improving the identification tests. Because as I mentioned, most labs really relied on the qPCR test proposed by Harper's and colleagues, which is really sensitive and really reliable as it is highly specific. But to go further in the identification of Xylella, especially in case of in samples infected by very low population sizes, we proposed a nested MLST to improve the typing step, but also to reduce the cost and the time used to detect and identify Xylella. We designed a one-step detection and identification assay based on the qPCR. And we evaluated quite recently the potential of a field test, the Amplify RP-XRT test that is proposed by ACDIA EMEA. And also we were associated to a work aiming at developing detection tool based on next generation sequencing. So just to quick slides to present the scheme that is used in France to detect Xylella in plant sample. So from plant material, after grinding, DNA is extracted and the reference method is the use of the quick pick test. And we rely so on the Harper qPCR test for positive and negative detection, for positive detection of the bacteria or for negative samples. And the subspecies identification is made either using these 2 genes, cis-G or MAL-F sequencing, or up to 7 housekeeping genes, where we need an entire sequence type determination in case of new area, for example. Then what we have to notice is that these MLST are based on conventional PCR and hence have a limited sensitivity compared to the qPCR test. And it is altogether a very long and expensive method of detection. We proposed some improvement to this method, especially to improve the level of detection, so the threshold, and to improve the identification. But this will not improve the time and the cost of this method. So we suggested to add a sonication step for the grinding. It is also proposed now in the standard published by the EPO, because this leads to the dislocation of the biofilm, so to a better access of the chemicals to the cells of Xylella, so a better extraction of the DNA. And we also proposed for some plant species for which the detection threshold was very poor, so for example, olive or oak, to use a CTAB extraction method. It's not a kit, it's more fastidious, but honestly, it's better. And we also propose to develop a multiplex. I will talk a little bit on that. And that help to detect and identify the bacterium in one step. And so we propose a nested MLST for improving the detection in case of lowly infected samples. So here I will just present the impact of the sonication and the CTAB in contrast to the use of quick pick and just grinding. So on 106 samples that were sampled in Corsica, and that represent 37 of plant species that include oaks and olive trees. The use of the classical methods led to obtain only 26 positive samples, whereas the use of the modified protocol, including a step of sonication and the use of CTAB extraction method, led to an improved of 300% as 80 samples were declared positive. So only these two modifications have a very strong importance for these samples for which detection level was very low. I should say that most CQ were above 25, and a lot of them was above 30. So the contamination of these samples was low. So I mentioned that for this low infected sample, we proposed to use a nested approach. Nested approach are very common in animal analysis for pathogenic bacterium, but also for foods, nutrients, because it's really improve the level of detection. So to be able to reuse the MLST database, that is very important, we have in mind to redesign the primers in order to have the same size fragment than the one used for the classical MLST sequencing. So my colleague Sophie, who redesigned this primer, modified a little bit the primer that were used in the Yuan et al. Primer, especially when we wanted to avoid high hairpin values and high differences in the temperature of melting between the forward and the reverse primer. And this was a problem for the GLTT and the All-C primers that were designed by Yuan and colleagues. So we redesigned these primers, and we of course designed primers that were exterior that were outer to these inner primers. And we tested the specificity through in silico and in vitro tests. And this was done also with the help of Blanca Landa from Spain, who sent us some material, both plant material and insect material. From France, we have access only to plant material. So here on this graph, you have the results in percentage of sequencing that was efficient using conventional or nested approach for each gene. So you can see that in most cases, the use of the nested approach allow an improved sequencing of all genes, and considering the use of the conventional or the nested approach for all the samples, so plant samples and insect samples in 2017 and 2018 from France and Spain, it is clear that most generally, the nested approach led to a very good improvement in the detection. It allows also to identify new alleles among French samples, and we were able also to identify recombinant and mixed infection using this nested approach. So I really recommend, as it is done officially in Spain, for samples that have low infection, so for samples for which CQ is above 30, to use the nested approach, but to be very careful and to avoid any cross-contamination, of course. So during a PhD thesis, Enora Dupas here developed a method to allow in one step the detection of Xylella and its identification at the subspecies level. So we rely on a robust approach involving in silico test and implanta test to validate the assay. So first, we look for specific sequences on 47 genome sequences, and we design primers and probe sets for various combinations. So either the species Xylella fastidiosa or for various subspecies, considering the subspecies Sandy and Morus, either within the fastidiosa subspecies or separately. Then we evaluate the performance criteria in simplex PCR assay on extracted DNA and bacterial suspension. We did multiplex primer and sprod in different combination and evaluate the performance criteria in multiplex qPCR assay and spiked and naturally infected plant materials. So we developed several sets of primers and probes that were specific, subspecies specific, and all were highly specific. I mean that no cross-amplification with any other subspecies or any other genera was recorded. And we used all the, in silico, at least all the NCBI database to do that. We also, in vitro or in planta, confirmed that these sets were highly specific. The qPCR assays are usable in tetraplex. This means that we could look for various subspecies in one area. The detection threshold that was obtained is as good as the Harper's one on pure culture. However, in plant matrices, it is equal or a little bit higher. But it is still interesting because it provides a subspecies identification at a level where the MLST doesn't work, because it is a qPCR assay here. So for CQs that are above 30, which means a bacterial concentration below 10 to the 4th or 10 to the 5th, depending on the plant sample, this scheme, this multiplex assay works for the identification of subspecies. And it is very cheaper than the current identification scheme because we don't need to sequence the genes for the MLST. So it is really cheaper. And it will be introduced in the 5th version of the EPO standard. So as I mentioned, I hope I am not too long, I haven't checked. We tested the potential of the ACDIA Amplify RP test. So first, this commercial recombinase polymerase amplification assays can be run directly in the field. There is no need to extract or to purify the DNA, and the kit operates directly on crushed plant material. This test targets a disulfide isomerase gene, which is present in only one copy in the genome sequences of Xylella fastidiosa. And we did check that by blastics across Xylella sequences and also across other bacterial sequences. And we had identity that were really lower for bacterium such as Xanthomonax, but which didn't result in cross-contamination. So these tests can be, well, this test can operate in two different ways, either in a real-time detection option that is completed using a portable device, or in addition, endpoint detection option is available. And in that option, the amplification is performed in a small heater block, and it is followed by detection using immunostrip reaction in a detection chamber. So we did a lot of analysis on plant material and strain, and we compared the results of this in-field test with data obtained using the use of the better test we have, the best test we have, so the qPCR of Harper. So on genomic DNA, the use of this RPA amplify test is quite good. At least it is highly specific. It detected all Xylella strain and non-target strain. We used 13 non-target strains and none was detected. So it is very specific. The limit of detection is not bad, but is not as good as the one of the Harper qPCR test. In fact, it is nearly 10 times lower for the Harper qPCR test. And in spiked plant matrices, again, it is highly selective. But then, depending on the plant species, the limit of detection can be really high. It is quite good for some species at 10 to the 3 bacteria per mil. So for almond tree, oleander, or curry plant, it's okay. But for some species such as grapevine, cherry tree, lemon tree, oak, or almond oak, and citrus, it is very poor. So we tended to analyze smaller volumes to dilute inhibition problems, and it improves the sensitivity. And then we wanted also to use the immunostrip detection to improve the detection when there is some inhibition. So we did that for the sample of almond and citrus, but also some samples of other oaks or lemon trees for which the detection was poor. And using the immunostrips for the detections give a very good improvement. So we did the same on infected plant samples that we have access to either after greenhouse tests or after natural sampling in Corsica area in France. And then when the infection was high, we have absolutely no problem. We could detect the infection using the amplifier portable fluorimeter. But when, or for some species, so for example, olive trees, the amplicon detection chamber did really improve the detection. So if you want to use this in-field option, you have to have in mind that the efficiency of this test really depends on the plant species you want to sample. And sometimes you will have to rely not only on the portable device, but also use the amplicon detection chamber, which will improve the cost of the detection. However, it's very quick and it's portable. and it is highly specific. And to finish my talk, I want to quickly present some of the work we were associated to, and aiming at using the possibilities offered by next generation sequencing to develop detection tools. So here we used the ISEC 100 sequencing capacities, and we compared it to really conventional detection methods. And here the objective of this test was really to develop to develop here the method of analyzing the data to be able to see how this sequencing can improve or not the detection and the identification at the subspecies level. So we did that on 27 field samples that were collected either in Italy, in France, or in the US, and they represent 6 different host species. So we had access to samples from olive tree, but also various ornamental plants, and oak and vitis, so vine, grapevine from the US. And what was interesting with that method is that running the sequencing is very easy. We rely on the preparation that was provided by the manufacturer. It is really easy. And then the analysis of the data set was quite also powerful, and we were able, for samples that were contaminated with very low population size, to have access to a few reads that allow us, in some cases, to identify the species. So here you have, for the various species, the samples and the contigs that were obtained. And the contigs that were obtained that are considered as specific for all the subspecies. So for all the samples that were declared clearly positive with a qPCR test, we have no problem. The NGS also detected very numerous reads that could be assembled in contigs, and it is clear that no problem, it can be used to detect. However, for some samples that were lowly contaminated, we had only access to a few reads. And even for one samples here of ornamental plants, so myrtle leaf microwort, very surprisingly, the qPCR test indicated a very low contamination, but the use of NGS ended in only a very few leads. This indicates that the bacterium was really aggregated in the sample. And here, what was interesting is that when we used distance-based ANI values of all these data that was recovered, we were able to identify or to assign the subspecies to those samples, even to those samples that were contaminated with only a few reads, except in these cases where the identification at the subspecies level was not possible, and it relies only to core Xylella reads. So I want to thank you all for your attention, and to thank all the colleagues that participated in this different part of the work. Thank you for your attention.
Thanks, Madge, for this excellent talk.
Thanks a lot.
Sorry?
Okay, I think I stopped the sharing of my screen.
Yes, please. Okay, thanks for this excellent talk. And the questions, just to remember that the questions will be in the end of the second talk. And also, I remember the audience that please write the question in the chat, and also fill the frequency form posted in the chat. For the second talk today, disease monitoring, you have a pleasure to have Dr. Maria Calderón Madrid as a speaker. Dr. Maria Madrid has a great experience in automated remote sensing methods for disease diagnosis. She holds degrees and her degrees in agriculture and civil engineering by University of Cordoba, and her PhD at the Spanish National Research Council focused on the early detection and verticillium wilt in olive using high-resolution spectral and thermal imagery. Later on, she's have been working on automate remote sensing methods for early detection of Xylella fastidiosa under the European project She's Actors, and continue with this work in the University of Salford that conducts a research project about optimization in early detection surveillance of Xylella by incorporating epidemic spread modeling with remote sensing imagery. Nowadays, she's working at Cornell University in the US, has a lead postdoc in a project building a global disease surveillance system for soil-borne plant pathogens. But she doesn't forget Xylella, and she's continues collaborating with the research project dealing with Xylella monitoring and using remote sensing. Maria, the floor is yours. You have 4 minutes, and good luck.
To the progress made until the date for the early detection of Xylella fastidiosa infection and monitoring of symptom development with the use of hyperspectral and thermal remote sensing imagery. Since the outbreak of Xylella fastidiosa was detected in Europe, in particular in Italy in 2013, the research conducted on the monitoring of Xylella fastidiosa with remote sensing followed 5 main objectives. The first one was to assess hyperspectral and thermal imaging methods for the early detection of Xylella fastidiosa-induced symptoms in olive. The second one was to evaluate spectral plant traits sensitive to Xylella fastidiosa across species, in particular olive and almond. The third one was to quantify the influence of the abiotic stress condition on the detection of Xylella fastidiosa. The fourth was to assess divergent biotic and abiotic spectral pathways to improve Xylella fastidiosa detection. The final one was the operational detection of Xylella fastidiosa using high-resolution commercial satellite multispectral images. Regarding the first objective, the question that arises is why it is important to detect Xylella fastidiosa infection at an early stage. Xylella fastidiosa is a bacterial plant pathogen transmitted by insects that feed on xylem sap. Xylella fastidiosa colonizes the xylem network within plants. The main function of xylem vessels is to transport water from plants' roots to leaves. So when the bacteria colonizes the plant, the xylem vessel becomes blocked and the plant slowly dies. To reach a new host, Xylella fastidiosa has to be carried and transmitted by a vector. However, once a plant is infected, it can take up to 14 months to start developing visible symptoms. Thus, asymptomatic infections or infections in early stage can easily escape surveys based solely on visual inspection. A recent article published by my colleagues in the United Kingdom demonstrated that the asymptomatic period for Xylella fastidiosa makes it very difficult to detect at an early stage when using visual inspections. This plot shows the relationship between the apparent prevalence and the true prevalence for a number of different pathogens, such as the one associated with the ash dieback, citrus canker, Juan Lombing, and the olive quick decline syndrome. The highest difference between apparent and true prevalence is seen for the olive quick decline syndrome caused by Xylella fastidiosa, highlighting the need for methods able to detect previsual infections which is particularly relevant given the threat of infected but asymptomatic trees contributing to the Xylella fastidiosa epidemics. The role of remote sensing in the early detection of Xylella fastidiosa is focused on the study of physiological changes related to Xylella fastidiosa infection across the different stages of disease development. For this purpose, we use the recent advances in imaging spectroscopy, a tool for the acquisition of images where for each spatial resolution element in the image, a spectrum of the energy arriving at the sensor is measured. These spectra are used to derive information based on the signature of the interaction of light with matter. Traditionally, remote sensing was based on the well-known three-dimensional data cube that resulted in the RGB color image, where each pixel has red, green, and blue color. Remote sensing has greatly developed in the last decade with the emergence of multispectral and hyperspectral sensors. The main difference between multispectral and hyperspectral imagery is the number of bands and how narrow the bands are. Multispectral imagery generally refers to 3 to 10 bands and is the data product delivered for the majority of the current commercial satellites. Meanwhile, hyperspectral imaging involves narrow and usually contiguous spectral bands. Application of hyperspectral imaging is limited to research due to the bunch of data generated to study physiological processes on vegetation, while multispectral imaging is more useful for commercial purposes. For the early detection of Xylella fastidiosa infection, we use high-resolution airborne hyperspectral imagery at sub-meter spatial resolution with over 250 spectral bands. This spatial resolution enables us to identify individual trees on the images by tree-crown segmentation algorithms and extract poor vegetation reflectance and radiance spectra to work with. The figure at the right bottom shows how the reflectance spectra changes as the severity of Xylella fastidiosa symptoms increases. We can see an increased reflectance on the visible region with the development of Xylella fastidiosa symptoms. Coming back to the previous slide where we hypothesized about the ability of remote sensing to detect physiological changes related to Xylella fastidiosa infection, I will explain the main physiological processes that change with Xylella fastidiosa infection and how remote sensing could detect them and at which stage of disease development. Structural changes are typical to Xylella fastidiosa infection, which consist in changes in the orientation of leaves in order to reduce the transpiration rate, leaf rolling, and eventually defoliation due to leaf ascission. These changes usually occur at advanced stages of disease development, so vegetation indices such as the popular NDVI are good indicators for damage mapping, not for early detection. Another physiological process that changes with Xylella fastidiosa infection is associated with the concentration of photosynthetic pigments in the leaf. Leaf discoloration is one of the most common and overlooked Xylella fastidiosa symptoms. The leaves turn yellow and to a brownish color. When this happens, it means that leaves are not receiving the appropriate nutrient and water supply. Thus, biochemical processes at leaf level break the chlorophyll down so that the green color disappears and the yellow to orange colors become visible through the development of carotenoid, anthocyanin, and xanthophyll pigments. These photosynthetic pigments differ from the way that they absorb incident light, making it possible to detect changes in the concentration with remote sensing from initial stages of Xylella fastidiosa infection. Another process that is disrupted due to the Xylella fastidiosa infection is the chlorophyll fluorescence emission. Maybe some of you are not familiar with this concept, which has attracted scientific attention in the last recent years. Incident light energy in leaves may be reflected, transmitted, or absorbed for photochemistry, thermal dissipation, or emitted to the atmosphere as chlorophyll fluorescence, which supposes to be a 2% of the total incoming radiation. Chlorophyll fluorescence is linked to photosynthesis efficiency in a way that when a plant is affected by a biotic stress and its photosynthetic efficiency decreased, the emission of chlorophyll fluorescence is also reduced. The lower graph shows the leaf fluorescence emission with maxima in the red and far red spectral regions. Chlorophyll fluorescence is emitted in narrow bands at 685 and 70 and 740 nanometers. So high spectral resolution sensors are required to its quantification. Solar-induced chlorophyll fluorescence has been demonstrated to be an early indicator for biotic stress in other studies dealing with vascular plant diseases. Lately, a reduction of the transpiration rate is caused by Xylella fastidiosa infection due to the block of xylem vessels. When a plant is infected by Xylella fastidiosa, it colonizes the xylem vessels, blocking the transport of nutrients and water from the roots to the leaves. The plant responds to its closing its stomata in order to reduce the loss of water with the transpiration. Thus, the evaporative cooling decreases and the ground temperature increases. We can detect this increase in temperature by analyzing thermal imaging, in particular the groundwater stress index, that is an index commonly used for detecting water stress in crops. Summarizing mapping of damage caused by Xylella fastidiosa, which consists in the detection of advanced stages of disease development, can be conducted by quantifying structural changes, photosynthetic pigments concentration, chlorophyll fluorescence emission, and transpiration. However, early disease detection is focused on the detection on detecting spectral differences between healthy and initially infected trees related to changes in photosynthetic pigment concentration, chlorophyll fluorescence emission, and transpiration. IRHS campaigns were conducted in Italy and Spain within the Xylella fastidiosa infected area, using hyperspectral sensor and thermal camera on board a main aircraft. Airborne campaign in Italy was conducted over 1,200 hectares within the demarcated infected area in the Puglia region in 2016 and 2017. In this campaign, we studied the early detection of Xylella fastidiosa subspecies pauca infection in olive trees. Another airborne campaign was conducted over 2,000 hectares in 2018 in Alicante province, central eastern Spain. This area is known because almond trees are naturally infected by Xylella fastidiosa subspecies multiplex. The last airborne campaign was carried out in 2019 and 2021 in Mallorca, Spain. To date, the Balearic Islands hold the major known genetic diversity of Xylella fastidiosa in Europe. Since October 2016, three subspecies, fastidiosa, multiplex, and pauca, have been identified infecting multiple host species, including grapevines, almond, and olives. Concurring airborne campaigns, field surveys were carried out to evaluate Xylella fastidiosa symptoms at tree level and sample plant tissue to confirm Xylella fastidiosa infection by qPCR. Moreover, physiological measurements were conducted at leaf level in relation to retrieved chlorophyll and flavonoids content, fluorescent emission, stomatal conductance, and reflectance spectra. These measures at leaf level will be used to better understand results obtained at imagery level. Once the reflectance spectra were extracted from each tree crown, the derivation of canopy structural parameters and leaf biochemical constituents was performed by inversion of the radiative transfer model ProSale. The model couples the leaf reflectance prospect model, accounting for leaf properties such as pigment concentration, and the canopy reflectance model SAIL, which accounts for canopy structural properties, such as the leaf inclination and the sound observer geometry. The inversion of radiative transfer models enables the simultaneous and independent retrieval of multiple leaf and canopy traits linked to physiological processes in plants. This approach is more likely to reveal the physiological processes associated with the disease and increases the potential transferability of findings to other datasets, diseases, and plant species. Nevertheless, there are specific narrow-band spectral indices that track processes currently not simulated by any radiative transfer model simulations, and therefore can complement these model estimated traits. Examples of these indices are the chlorophyll fluorescence and the crop water stress index, among others. Then we use classification and machine learning algorithms fed we use machine learning algorithms fed by this pool of functional plant traits to classify disease incidence and severity. Sensitivity analysis of plant traits of Xylella fastidiosa infection showed that the chlorophyll degradation trait, it is the MPQI index, calculated in the blue region, and the crop water stress index best distinguish Xylella fastidiosa symptomatic from asymptomatic trees, followed by anthocyanin, carotenoids, and chlorophyll fluorescence. The importance of the functional traits varied as a function of Xylella fastidiosa symptom severity, so that MPQI and crop water stress index most reliably distinguish symptomatic from asymptomatic trees, but were of lesser importance to discriminate between initial and advanced stages of disease. For these symptomatic trees, chlorophyll fluorescence was the most sensitive functional trait to detect low severity of Xylella fastidiosa symptoms. The left figure shows the validation of the remote sensing model with qPCR data. It enabled the generation of a spatial map of disease incident prediction by remote sensing, revealing infected asymptomatic trees that were missed by the visual evaluations but detected by remote sensing. The accuracy of the remote sensing model validated with the tree-level qPCR dataset yield an overall accuracy of 94%, whereas the performance of visual inspection against qPCR was of 78%. Moreover, the remote sensing model detected up to 90% of the infected asymptomatic trees that were missed by visual assessment. It demonstrated the ability of remote sensing to pre-visual detect Xylella fastidiosa infection, overcoming traditional scouting methods based on visual inspection of symptoms at leaf level. Jumping into the second objective, here this figure shows the normalized importance of hyperspectral and thermal plant traits to detect Xylella fastidiosa symptoms across olive and almond trees. This approach revealed how important individual spectral traits were in the overall response of the two host species to Xylella fastidiosa-induced stress. Crop water stress index, fluorescent emission, and the xanthophyll-related spectral traits, PRI, are common indicators of Xylella fastidiosa in olive and almond. MPQI and anthocyanins are olive-specific, while chlorophyll and the leaf area index are almond-specific. In the third study, we disentangled biotic stress caused by vascular xylem invading pathogens from abiotic stress imposed by water limitation by revealing distinct spectral pathways associated with the physiological alterations detected through mapping spectroscopy and thermal data. This is of paramount importance for large-scale remote sensing detection efforts of early disease infection symptoms that occur in the context of natural physiology variability due to the presence of abiotic stressors in agroecosystems. This figure shows the normalized importance of spectral traits for Xylella fastidiosa detection across species and the simultaneous biotic and abiotic stress. We used the specific spectral traits linked to the biotic stress imposed by Xylella fastidiosa infection to determine for each pathosystem which indicators deviated from the abiotic stress response. Crop water stress index resulted in to be a common indicator of Xylella fastidiosa and water stress. The chlorophyll fluorescence emission is important for Xylella fastidiosa detection across olive and almond. MPQI and anthocyanins are olive-specific indicators for Xylella fastidiosa infection, while PRI and carotenoids are almond-specific indicators for Xylella fastidiosa infection. In the fourth objective, we evaluated if Xylella fastidiosa-associated biotic-abiotic spectral fingerprints were distinct from those detected for Verticillium dahliae. Verticillium dahliae is a xylem-invading fungus that causes verticillium wilt, the most devastating soil-borne disease infecting olive trees worldwide. Notably, Xylella fastidiosa and Verticillium dahliae are two distinct vascular pathogens that increase resistance and eventually block water flow through the vascular system, triggering similar symptoms in olive trees. This collapse in water flow reduces transpiration and induces water stress, thus causing analogous symptoms that also can be confounded with abiotic stress. In addition to the airborne campaign conducted for Xylella fastidiosa infected area in the southern Italy, we scanned Verticillium dahliae infected areas in southern Spain between 2011 and 2013. This figure shows the normalized importance of spectral plant traits for Xylella fastidiosa and Verticillium dahliae detection in olive trees under simultaneous biotic and abiotic stress. We demonstrated again that the crop water stress index is a common indicator for Xylella fastidiosa and Verticillium dahliae biotic stressors, as well as for water stress. MPQI, anthocyanins, and fluorescence emission are divergent indicators between biotic and abiotic stress. But PRI and the blue index via F are Xylella fastidiosa specific divergent indicator from Verticillium dahliae. And carotenoids, blue index, and the leaf inclination distribution function are Verticillium dahliae specific divergent indicators from Xylella fastidiosa. Accounting for abiotic, biotic, and species-divergent plant traits greatly improve model performance for all datasets comprising both species and both pathogens. Model accuracies for Xylella fastidiosa in almond reach overall accuracies of 94%, up from the original 83%, while we achieved overall accuracies of 92%, up from the original 62% in olive trees. In the case of Verticillium dahliae infected trees, we achieved overall accuracy of 93%, up from the original overall accuracy of 75%. Okay. For the last objective, we studied the impact of using high-resolution commercial satellite imagery for the detection of Xylella fastidiosa, comparing against high-resolution hyperspectral and thermal imagery as a benchmark. This figure shows a comparison of the spatial and spectral resolution between narrowband hyperspectral images and world-view 3 multispectral images. Airborne hyperspectral images have a pixel size of 0.29 meters and were collected consisting of 360 71 bands in the spectral range from 400 and 1000 nanometers, while satellite multispectral images have a pixel size of 1.24 meters and were collected consisting of 8 bands in the spectral range from 400 to 900 nanometers. These figures show the accuracy results and the first positive and negative rates when applying our model to the satellite data with groundwater stress index, airborne hyperspectral data without using groundwater stress index, and hyperspectral data with groundwater stress index and chlorophyll fluorescence. Accuracies from satellite data were considerably lower when compared with the airborne hyperspectral benchmark dataset for detection of early, middle, and all stages of the infection. These results improve when adding the thermal groundwater stress indicator, but they are still so much lower when comparing to the hyperspectral ones. Regarding the figure of false positive and false negative rates, we realized that the false negative rates were so high for satellite data, which means that a lot of symptomatic trees were classified as healthy trees. Thus, the next step was studying why this high rate of false negative is occurring when using satellite images as compared to airborne hyperspectral data. Based on this, we performed a tree-based prediction of a large area to detect Xylella fastidiosa infected olive trees. The left figure shows that the areas predicted as infected by the models built with hyperspectral data were larger compared with the areas predicted by the satellite. Then we plot the relationship between the proportion of trees predicted as asymptomatic using the only hyperspectral information with the proportion of trees predicted asymptomatic using the only satellite multispectral information. The proportion of infected trees detected as asymptomatic was in all cases higher with the airborne hyperspectral model than with the satellite data, observing plots with large errors, while others were accurately estimated. The question that arises now is why some plots show large errors between prediction from satellite and hyperspectral data, while others don't. For the plots where this discrepancy was the highest, the proportion of trees at early stages was higher for all cases, while trees at advanced stages were not observed. On the other hand, those plots with the lowest discrepancies had the smallest proportion of trees at early stages and the highest proportion of trees at advanced stages. This analysis demonstrated that the major discrepancies between satellite and airborne hyperspectral were generally observed when trees at early stages are present, while the greatest similarity was obtained where more advanced stages of the infection and less presence of early stages were observed. The relationship between symptomatic trees detected by satellite and airborne hyperspectral against the proportion of trees at the early infection stage showed a positive trend, suggesting that the main driver of the error obtained from the main error was obtained due to the percentage of trees at an early stage of infection. This study suggests that the use of lower spatial and spectral resolutions from satellite imagery prevents us from the early detection of Xylella fastidiosa infection at landscape level. So the final conclusion for the progress made up to the moment in the detection and monitoring of Xylella fastidiosa in the European Union was first that the progress made on assessing hyperspectral and thermal remote sensing for Xylella fastidiosa infection across olive and almond species obtained overall accuracies up to 80-90% and kappa values over 0.6. The second conclusion was that remote sensing overcomes traditional scouting methods based on visual inspection due to its capability of detecting previsual symptoms and screening large number of hosts in a short space of time. The third conclusion is that specific spectral plant traits have been identified to successfully detect Xylella fastidiosa induced symptoms in olive and almond species. These indicators were groundwater stress index, NPQI, anthocyanins, chlorophyll fluorescence, and the xanthophyll-related index, PRI. The fourth conclusion was that quantifying divergent spectral traits for abiotic and biotic stress is critical for improved Xylella fastidiosa detection in almond and olive. And the final conclusion is that a high-resolution commercial satellite multispectral images are suitable for Xylella fastidiosa damage mapping, while early detection requires hyperspectral data. Before finishing my presentation, I would like to acknowledge the contribution to this work of my international colleagues from the Joint Research Center in Ispra, Italy, Instituto de Agricultura Sostenible in Córdoba, Spain, University of Melbourne in Australia, and Swansea University in United Kingdom. Thank you very much for your attention.
Thanks, Maria, for this excellent talk. It's interesting. Now you are open for the questions and answer sections. Please, both Marias, go open the camera. So we have some couple of the questions for Maria, Jacques, and after to Maria, Madrid. The first one that you was sent us, just a moment, please. Okay. Mad, the question was with Mariana Bossi. She's asking about the mechanical inoculation by the NID method in olive citrus plants. The question is, how long after it's possible to detect the bacteria by molecular tools? Do you understand the question?
Yes, thank you very much for this question. So I would say that it depends a lot on the condition in which you incubate the plant, and it depends also a lot on the strain. So I mean the subspecies you are testing on these various hosts. So in our condition, so in France, Xylella is a quarantine pathogen. So when we are doing some mechanical inoculation, we have to do that in confined host chamber, which is a very artificialized environment. And in this kind of environment, we never were able to see any symptoms on olive plants. And we made only a few experiments with citrus. We did show some symptoms on plum, but for all plants, when the combination, so host species and subspecies of Xylella were favorable, we were always able to detect the bacteria, at least at the inoculation point. The bacteria did survive very well at the inoculation point. And then quite soon after the inoculation, we can follow the bacteria by qPCR a little bit above the inoculation point for all plants we inoculated. Again, when the combination is favorable. For example, if you inoculate fastidiosa, so Xylella fastidiosa strains on olive trees, soon the detection at the inoculation point will be poor and the bacterium will disappear. But in any other case, when it's only a mean of using molecular technique to detect the bacterium, it is quite efficient.
Okay, so just for understanding, Maria, when for example, using the inoculation, for example, in citrus, if you inoculate the subspecies fastidiosa, you just found the bacteria inoculation point.
Okay. Well, I cannot understand. I cannot answer that because I never did that. I never did fastidiosa on citrus. We did a lot of inoculation in plum, in ornamentals, a lot of inoculation on olive plants and grapevine using the various subspecies, but only a very few on citrus. It was to test Pauca strains. Because for us, the conditions we have in our growth chamber are not very favorable for citrus plant. Yeah. So I'm not very able to answer for citrus plant.
Okay, no, sure. Just what was I use in citrus just for an example, but I mean the for example in multiplex or in olive plant, for example, using multiplex or fastidiosa subspecies. When you inoculate, the bacteria is easy to be found in the point of the inoculation. But after that, how long time you can find, or how long, or you can find the bacteria through the plant?
You understand? In upper part of the plant?
Yeah, the top of part of the plant.
Yes, for us, the easiest plant to manipulate is myrtle leaf milkwort, so an ornamental, Polygala myrtifolia. And for Polygala myrtifolia, in less than 2 weeks, we were able to detect the bacterium at least 20 centimeters above the inoculation point. And vitis inoculated with fastidiosa strains are very easy to run plants. For all other, it is much more complicated.
Yep. Okay. Yeah, I know. I know.
Especially olive trees. And you know that well also.
Okay. The second question is from Maria Gabriela dos Santos. The question is if you have some information about the contamination using pruning. Scissors by pruning. Do you have some information about this one?
No, I have absolutely no information. We have never tested that. And in fact, for us, it is not possible to test that because we are in a situation where every time a plant is declared infected, we have to remove the plant very quickly. So we never are able to detect any dissemination on that plant. So it is very limited environmental conditions to detect that. But I would be very interested to see if it's really impossible to detect the transmission of the bacterium by pruning shears, because it is not obvious that it is not possible. But I remember having read old papers that show that it is not efficient at all. So I would be very interesting to see if it's common or not, but apparently it's not common. It's not efficient. But we were not able to test that.
Okay, thanks. Just for learn a little bit more about from you, well, I have one question. Okay, when you mentioned that the using the I can remember the sonication in the samples, the tissue. The matrix was crushed or just sliced?
No, we crushed the material and then we sonicated it. And really the impact on the extraction of DNA is really interesting. And especially for those samples which have CQs above 30.
Okay, cool. The last one, it's about the CT value. The threshold. Okay, do you, if I remember, in some slides, I see some CT value between 38, 39, something like this one. Did you consider this CT value positive or negative? Do you have the question is, do you have a threshold that was standardized for all the hosts?
Yes, for us in France, officially at least, the CQ was up to 2017 certified to discriminate between positive and negative sample. And then afterward, officially, the CQ was elevated at 38. So 38 is the limit between positive and negative samples. And in fact, for us, but we are not an official lab, we are doing research, we always saw that when we are sampling naturally infected samples, if we made a distribution of the CQs, in fact, naturally, you have a gap around 38, and you have a quite regular distribution up to 38, and then you have nothing. So it is what is written in the paper written by Harper. 38 is the threshold to use. It can depend on hosts, but for us, at least for the host we sampled in France, apparently it doesn't play any role. So 38 is now the threshold we use.
Okay.
And it is very uncommon to see any plant or any plant material that is infected above 38, except if we are adding a lot of concentration steps or any other step in the extraction method that could modify a little bit this data.
Okay. Thank you very much, Maria, for these answers. So the next question will be to Maria from Spain. Okay, Maria, the question is about, does it possible to you observe one pattern of distribution, I mean, randomly aggregated or linear distribution using these images? I think this, do you understand it?
Answer is what for me. Can you repeat, please?
Okay. This is possible to observe a pattern of the distribution of the plants by the image. I mean, like randomly linear distribution or aggregated distribution.
Yeah, of course. Indeed, for example, the most easier thing to do with remote sensing is to map the damage. And the damage, finally, we know that this type of diseases that they are transmitted by vectors, the spatial distribution is in clusters.
Okay.
It's like other soil-borne pathogens like verticillium. So we can see the cluster clearly in the field when we are mapping the damage. Even it's better the mapping when we are trying to detect early symptoms with hyperspectral airborne imagery, so we can see better this pattern. And also during my PhD in Salford, we were trying to feed epidemiological models with remote sensing imagery in order to see how this pattern spread temporally and especially in time. So we saw that, for example, a remote sensing data is a better source of data than visual inspection for parameterize this type of models.
Okay. Another question is about it's not a question, but it's a curiosity. When you saw the work that was done in Italy using the airplane, what? Okay.
I don't understand you. What?
Okay. When, if I remember from your talk, when the work that it was done in Italy,
it was in 2016 and 2017.
Yes, but it was using airplane.
Airplane, yeah.
Yes.
Airplane with hyperspectral and thermal sensors.
Okay, but my question is about the VNT. Is it possible to use VNT also?
VNT?
Yes. It's it's a drone.
The drone.
Yeah.
The problem of drones, like they only cover a small area. So, for example, the restriction is almost 10 hectares.
Okay.
And for this type of, for example, for Xylella fastidiosa, we use aircraft because we can cover a huge area of space in 2 or 3 hours. So it is better than drones. Drones, I think that now that they are very trendy in the world of remote sensing, it is only suitable, for example, for plot purposes, only for a plot or two.
Okay, but okay, I understand you. Another question is this about the. I imagine that for each host or each country, you must standardize one typical wave, the typical waves that you obtain. Okay.
Yeah.
Okay. The question is, if, for example, in the almond. I know. My question. I don't know about this one, but is using the rootstock in almond. If using the rootstock could be have some influence in the patterns that you can read.
I'm not specialist on it now.
Okay.
So I only, because I still working on it, but when I went to the field, it was only in the olive area in Xylella fastidiosa in Puglia region. But my other colleagues were scouting the symptoms in Alicante, in Almos, and in Mallorca, because I went on my postdoc here, okay. So it's very far. So I couldn't see it, so I have no answer for it. I don't know.
No, I just asking you about this one because, for example, in citrus, using different kind of the rootstock.
Yeah.
That's varieties, and you see the naturally not related with the disease, but naturally you have some difference of greens in the leaves according with the rootstock.
Yeah.
And okay, so that was I thinking about this question.
Yeah, the problem is that the almond area that is infected in Alicante, it is not commercial. They are not commercial almonds. Okay. They are rain-fed almonds. So I think that the people is not studying it because these almonds they have been there for a long time and they have a very low productivity. So it's not like an issue to study. I think so. I don't know.
Okay, thanks. The last question from Samir Akhtar. It's the question: is there any data regarding the geographical distribution of this bacterium? I don't know. I can't understand very well if it is from it's for you and Maria.
Distribution of this
yes.
bacteria. I think that there is. We try, for example, in the European Union to have updated data about how is the spread of Xylella fastidiosa in the territory, but we don't know exactly how it is spreading because we know that it's very difficult to detect because of the long asymptomatic period. But another colleagues from Spain, they are working on modeling how the environmental conditions they are conducive to the development of symptoms of Xylella fastidiosa, and we know where are the areas that they are conducive for the spread of this disease, but we don't know a lot, or we don't know exactly where is the bacteria. We know where it is the outbreaks, but now we are doing a lot of monitoring and surveillance to see if one plant is introducing in a new place, but it is very different to control. It's very difficult.
Okay, so the last questions will be from Maria Jacques for you. Okay, are they able to differentiate almond trees infected by different subspecies in Mallorca using this hypothermal imaging?
If it's not clear, I can add some comments.
Okay, sure.
Thanks a lot for your talk. It was very nice. I wondered if in Mallorca you can have access to almond trees that were infected by fastidiosa strains, but also by the multiplex strain. And if your hyperthermal imaging can just tell you that these almond trees are infected, which is very good. Or if you have any physiological answers, as you demonstrated, the presence of anthocyanin or flux or chlorophyll or anything else that tell you maybe there is a difference in the subspecies invading these plants?
We didn't try it yet, but if I don't know a lot about it, but if they have specific symptoms, I don't know if they have specific symptoms due to the subspecie, it could be able to be detected. But if not, I think it is very difficult to detect the subspecie that is infecting almond trees.
Okay. Thank you.
Thanks. So if you know more questions, the organizing committee is grateful for both speakers, excellent talks and discussion, and also the excellent contribution, and also you are grateful for all of the listeners. So just remember, the next week you're going to have another section about the management of disease in Brazil, US, and Italy. That will start in 11 a.m. So we waiting for you the next week, and have a great weekend. Thanks.